Hello, my name is Thomas Verstraeten and it is my pleasure to talk to you about the design and recruitment of Phase 3 vaccine trials. As the title of this course suggests, I will focus on trials of vaccines and what may be different in such vaccine trials compared to trials of the more traditional drugs. I will assume that you know the difference between Phase 1, Phase 2 and Phase 3 trials. I will start my lecture with a brief overview of the typical endpoints of Phase 3 vaccine trials. I will then spend some time on a number of key elements that need to be decided when designing a Phase 3 vaccine trial. Next, I will discuss some recruitment topics, and finally I will conclude by proposing some further reading on Phase 3 trials of recently licensed vaccines. The single most important aspect of any clinical trial, and certainly a Phase 3 trial, is determining the primary endpoint. This is critical as the success of the trial, and in a way of the entire clinical development program of a new vaccine, will depend on whether the Phase 3 trial will meet its primary objective. This primary objective is nothing else than a statement on the expected level that will be achieved of the primary endpoint. Typically for a Phase 3 trial, this will be an efficacy endpoint, in most cases simply the vaccine efficacy. Whereas this may seem very straightforward, we will see later in this lecture how the details of the endpoint may require some very careful consideration. A primary endpoint may also be related to safety. Not infrequently, such an endpoint will be a so-called co-primary endpoint to an efficacy endpoint. The immunogenicity of a vaccine is of course a very important measure of a vaccine's capacity to mimic a natural infection, and will most typically have been measured in Phase 1 or Phase 2 trials. In some circumstances, where the outcome to be prevented cannot be measured because it is too rare or because it is already being prevented by another existing vaccine, as may be the case for polio for example, this immunogenicity may be the primary endpoint of a Phase 3 trial. As you will have learned, many vaccines not only prevent disease in those vaccinated, but also in those unvaccinated, an effect referred to as the indirect effect of vaccination or herd protection. In some Phase 3 trials, though this may be more typically measured in later so-called Phase 4 trials, this indirect effect may be the primary objective. Besides the primary endpoints, a Phase 3 trial will typically also have a number of secondary endpoints. Most typically, the immunogenicity of the vaccine will be measured in a subset of the participants. Safety, if not a primary objective, will always be a secondary objective. Not infrequently, efficacy may be measured in certain predefined subgroups, will be measured for different endpoints than the primary one, and, typically for vaccines that require multiple doses to achieve optimal protection, efficacy will also be measured by the number of doses administered, for as far as possible. I would now like to go into a bit more detail on a number of aspects of a Phase 3 trial that, in my experience, have often led to long and sometimes heated debates in the groups that are designing these Phase 3 trials. Nearly all of these points can lead to the success or failure of the trial, and thus in a way to the success of the entire development program, the vaccine under development, and even in some instances the success of the company developing the vaccine. As mentioned before, the definition of the primary efficacy endpoint is certainly the single most critical aspect of a Phase 3 efficacy trial. The actual disease that makes up the endpoint is usually obvious, but as often in medicine, the way a case of the disease is defined may vary a lot. Some diseases that are notorious for having multiple case definitions are, for example, pneumonia and acute gastroenteritis. Different case definitions will have different sensitivity and specificity. This may lead to difficult choices on whether to choose for the most sensitive case definition and thus provide the most power to the study, or whether to choose for high specificity and thus ensure the highest possible efficacy can be achieved. Many infectious diseases will result in different disease syndromes with differing levels of severity. The most obvious difference is whether the disease has led, for example, to a hospitalization or not. For some diseases, specific severity measures have been developed to make the distinction between severe and non-severe disease more objective. As some vaccines, such as the rotavirus vaccine, have been shown to protect primarily against severe disease, the choice of the severity to be measured may again determine the chance of success. Related to the above points is of course the choice of diagnostic tests to be used. Not infrequently, existing tests may not be sufficiently well established or validated for the purpose of a phase 3 trial and will be developed for the specific purpose of the trial. For some endpoints which are not so severe and may not always lead to a visit to a healthcare professional, cases may need to be ascertained proactively by, for example, visiting the home of the vaccinees every week. This will of course lead to a large financial and logistical resource needs and are therefore to be carefully considered. Finally, for some outcomes that take too long to develop, alternative endpoints, often referred to as surrogate endpoints, may be proposed. Let us turn now to another important element, which is the choice of the best control to which a new vaccine will be compared to in a phase 3 trial. Whereas from a scientific perspective a placebo injection may be the best option, this is not accepted as ethical in many regions in the world. Another vaccine will therefore need to be chosen, but this poses many challenges. First of all, this vaccine must be licensed in the countries where the trial will take place, but not recommended, as it must otherwise be given to both groups. In addition, the control vaccine should not have any influence on the efficacy endpoints and have a comparable safety profile to the vaccine under investigation. In large multi-country trials, this choice becomes particularly difficult and may actually lead to certain countries being excluded from trials. In clinical trials prior to phase 3, the experimental vaccine has most likely been administered in the absence of any other vaccine, thus without simultaneous co-administration. However, for the vaccine to be later recommended in a routine vaccination program, where other vaccines are likely to be already recommended to the same target age group, the influence of other vaccines on its immunogenicity and efficacy will need to be tested. Sponsors may wish to already test the impact of co-administered vaccines during the phase 3 trials. This needs to be balanced against the desire to achieve an optimal vaccine efficacy without potential interference of the other vaccines. In trials involving infants, it is almost impossible to avoid co-administration of other vaccines, unless sponsors opt for administering the new vaccine outside the recommended schedule, in a so-called staggered scheme. Similar to the choice of the control vaccine, the choice of vaccines to be co-administered with the experimental vaccine is often a difficult decision, with differences in vaccine schedules between countries to be taken into account. The choice of the randomization ratio, that is the ratio of the number of subjects receiving the new or experimental vaccine compared to the number of subjects receiving the control, is usually based on practical considerations. Classically, this is a 1-to-1 ratio. One consideration, however, may be to expose more subjects to the experimental vaccine so as to enlarge the so-called safety database. The safety database is the total number of subjects exposed to the new vaccine for which safety data are available. In trials with rare efficacy endpoints, the number of subjects receiving the new vaccine will usually be sufficiently large to ensure a safety database large enough to detect rare adverse events. But in situations where the overall number of subjects exposed to the new vaccine is not so large, there may be a need to ensure more subjects are exposed to the new vaccine. The risk of using a so-called unbalanced randomization ratio, for example a 2-to-1 or a 3-to-1 ratio, is that rare events are more likely to be detected in the group of subjects receiving the new vaccine. compared to the control group. Although this will rarely create a statistically significant imbalance between the groups, it will create some uncertainty on whether the events can be attributed to the new vaccine. This may result, in the worst case, in a halt to the trial, or in other cases in large post-licensure safety commitments. The most suited vaccine schedule is usually based upon immunogenicity results, but this does not necessarily mean that this also represents the schedule with the best efficacy results. Different vaccine schedules may therefore be measured in a phase 3 trial, although alternative schedules will more frequently be tested in post-licensure phase 4 trials. More likely will be the testing of the effect of a booster dose, as the need for a booster dose will often not be known until years after the first efficacy measures become available. Studies will then be extended to allow the subjects to be re-invited to receive a booster vaccination. To measure the effect of a booster dose at different time intervals, the original group of trial subjects may be divided into different groups to be invited at different time periods after the original vaccination. Another question that arises is whether the subjects who did not receive the experimental vaccine should receive it at the end of the trial. This is of course only the case if the vaccine was found to be efficacious. Whereas this seems like a logical choice to be made, it does limit the possibility of studying the long-term effects of the new vaccine in a controlled fashion. This can still be measured by measuring the rate of vaccine failures in the exposed group, but of course this will no longer be a randomized experiment. An alternative solution that is sometimes proposed is to offer the trial subjects a so-called beneficial vaccine, which is another vaccine that has been shown to be beneficious and efficacious, but is not routinely provided in the countries where the trial takes place. The duration of the follow-up of the study subjects is again a balance between a desire to bring a new vaccine as soon as possible to the market to help meet an unmet medical need, and the desire to study its long-term effects before its use in the real world. The long-term effect of a vaccine may be suspected based on immunogenicity data obtained from long-term follow-up of subjects in phase 2 trials. Frequently, sponsors of new vaccines will be required to provide long-term follow-up data of the trial subjects on a regular basis to substantiate the expectation that long-term immunogenicity will also result in long-term efficacy. In certain study populations like military personnel or travelers, long-term follow-up may not be an option, and observational studies may be required in the post-licensure setting to substantiate the long-term protective effects of the vaccine. It is an obligation of the study sponsor to collect safety information on every subject that is enrolled in a clinical trial, and this is certainly not different in a phase 3 trial. Typically, the safety information that is collected is categorized as either serious adverse events, or SAEs, pregnancy outcomes, unsolicited adverse events, and typically for vaccines, a range of local and general solicited adverse events. A category that is almost routinely included in vaccine trials are the so-called adverse events of interest. Most often, these will be immune-mediated events, including allergic reactions and autoimmune events. In addition, if safety signals have been detected earlier in the development program, these may warrant special attention as well. Note that phase 3 clinical trial will almost always have a data safety monitoring board, or also called an IDMC, to oversee the safety of the subjects during the conduct of the trial. The choice of the countries in which to conduct a phase 3 trial needs to be made carefully. Most companies acknowledge that they will only conduct trials in those countries where they also have an intention to license and market the vaccine if approved. We already mentioned the need to ensure that all recommended vaccines are given to the study participants, often in co-administration with the new vaccine. The choice of countries is most frequently determined by the experience of investigators to conduct large phase 3 trials, the expected difficulties in obtaining approval from the regulatory authorities, and of course the need to include subjects from countries that represent the populations in which licensure will be sought. As a consequence, a few countries or regions have become very popular with vaccine manufacturers, given the large experience of the investigators and authorities to conduct these trials. These include Finland, several Latin American countries and the United States. Finland and other Scandinavian countries are of special interest, as subjects can often be followed up for a long time after the trial has finished using existing population healthcare databases. Given the large sample size of many phase 3 vaccine trials, they will often be conducted across several sites and several countries. Recruitment of subjects in a phase 3 vaccine clinical trial is probably not very different from recruitment into a classical drug trial, with the exception of the sometimes very large number of subjects to be included. There are a number of special considerations that may need to be made. For example, there might be a desire to stagger the enrollment over different age groups if the safety database prior to the start of the trial is too small to immediately vaccinate small children. In this case, an IDMC will frequently be requested to review the safety data in the first age group before starting to vaccinate the younger age group. For some infectious disease with marked seasonality, there may be an interest to ensure recruitment start before the actual season. Generally speaking, there is an interest to recruit over a relatively short period to avoid large variations in the frequency of both the infections to be prevented and the adverse event reporting. For logistical reasons, this will also facilitating moving from one stage into another stage in a harmonized fashion across different sites. Not infrequently, the sponsor will stimulate recruitment by allowing so-called competitive enrollment. In this case, the number of subjects to be enrolled at each site is not predetermined, but dependent by the capacity of each investigator to recruit rapidly. In and exclusion criteria need to be defined for every trial, but are particularly critical for phase 3 trials as they will directly influence the labeling of the vaccine if approved. Generally speaking, authorities will only grant a license for those populations included in the trials. In practice, it is of course not possible to ensure every population group is represented in the trial, but some groups will require special attention. These groups are, for example, subjects with chronic illness who may have been enrolled if they are not in an active state of their illness. In the case of infants, that may be children born prematurely. Certain racial or ethnic minorities may constitute special subpopulations in a trial. In countries with high HIV prevalence, subjects with HIV infection may also constitute a subpopulation of the trial. For every phase 3 trial, there will be a large team of personnel available to support the investigators to conduct the trial as efficiently as possible. Nonetheless, the motivation of the study staff at the sites is a critical element, especially in trials that have a long duration. Some relatively simple communication tools such as a newsletter can be a very effective tool in maintaining the staff's motivation high. The sponsor of the study will keep a very close eye on the enrollment progress and measure this against well-defined and predetermined targets. The comparison of enrollment progress will be particularly informative to understand the factors that drive or impede enrollment. Almost as important as enrollment and often seen as an important measure of a site's capacity to conduct a trial is the so-called attrition rate. This represents the proportion of subjects that drops off during the trial and will be representative of the staff's capacity to motivate the subjects or their parents to follow the study procedures throughout the trial. A particular challenge in many countries for conducting trial involving children is the requirement to obtain consent from both parents, especially if the involvement of both parents in the care of the child is not the local norm. Finally, I would like to suggest a few topics for further reading. The first vaccine I'd like to suggest for further reading is the HPV vaccine. Currently available HPV vaccines were licensed based on a surrogate endpoint. The most important disease to be prevented by this vaccine is cervical cancer. However, as it takes very long, up to decades, to move from a HPV infection to cervical cancer, it was not practical to use this disease as an endpoint in clinical trials. Instead, vaccine manufacturers have studied the effect of HPV vaccines on precancerous lesions as a surrogate endpoint. The next vaccine I would like to highlight is the rotavirus vaccine. As the first licensed rotavirus vaccine was shown to be associated with intussusception, a rare but severe disorder of the bowels in small children, all subsequent candidate rotavirus vaccines had to show the absence of such a risk in their clinical development program. As a consequence, manufacturers had to conduct very large phase 3 trials across several countries without any prior knowledge on the potential association of their vaccine with this rare outcome. Finally, I'd like to highlight a huge phase 3 trial conducted by one manufacturer to show the superiority of an adjuvanted seasonal flu vaccine over a non-adjuvanted seasonal flu vaccine. This trial failed to meet its primary objective, and as a consequence, the manufacturer stopped the development of this adjuvanted vaccine. It is an illustration on how even in the vaccine world, success is not always guaranteed, even if the basic science and phase 2 trials are very suggestive that a vaccine will meet its targets. I'd like to thank you for your attention for this lecture and wish you a good continuation in the course.